Okay, welcome to Mycorrhizas. This is the YouTube channel for the South American Mycorrhizal Research Network. My name is Jack Scher. I'm a PhD student at Northern Arizona University, and today I have the pleasure of speaking with Justin Stewart, who is a quantitative ecologist at the Society for the Protection of Underground Networks, aka SPUN, and we're going to talk about his work mapping the biogeography of arbuscular mycorrhizal fungi around the world. Justin is the lead author on a recent paper called The Global Density and Biomass of Arbuscular Mycorrhizal Fungal Networks, which was published in Science just a couple weeks ago. So thank you for joining us, Justin.
Thanks for having me, Jack. I'm always excited to talk about fungi.
Yeah. All right. So before we kind of get into the bulk of the interview, Justin has a couple of visual aids to kind of introduce this concept of mycorrhizal networks, as well as share a bit of background about how this work was done. So, Justin, do you want to share your screen?
Sure. Yep, it looks like you can see my screen. So, I find it helpful to frame the conversation around this project that we did, and I hope we can get more into the details in a moment. But I always like to start all of these interviews with this video or one of the videos. It depends. As you can see my tabs at the top, I have two. Um, So what we're looking at here are hyphae of arbuscular mycorrhizal fungi, and I know the audience here knows this group of fungi I'm talking about, but 70% of land plants, 450 million year old symbiosis, and they're direct points of entry of carbon from the atmosphere into soils. And so this video taken by my colleague Victoria Terry at the Amhof Biophysics Institute in Amsterdam is showing the real-time flow of carbon from a plant into fungal networks flowing from the part with the root down to the hyphal tip. This is not sped up. This is in real time. So these are the types of flows that are happening underneath our feet. With this study, we asked a very basic question, just how many of these networks are there on Earth? So we figured out that there was actually quite a big problem. It's kind of like saying, that there's 100 million trucks going across Earth roads all year, or every day, but you have no idea what is the type of infrastructure required to complete that resource transfer. So this study set to figure out how much of this carbon is going into Earth soils, building hyphal networks, and importantly, where are they located? So moving from this micron scale, which in my view is the most beautiful part of this study, is we've merged micron scale work with global modeling. I want to show what has actually come out of that. And so this is I think we can maybe put the link to this in the caption, but we've built an interactive web app where you're able to go and zoom in and look at our types of data we've been generating. So this was made by first of all the scientific publication, but designed with Moritz Steffener, who is a beautiful award-winning data visualization artist. Is visualizer a word?
Yeah.
And what we have here is a map showing the global distribution of hyphal density. So I'm going to zoom into actually right outside of my grandmother's house and explain a bit about what this study did. So I'm going to leave it here. My grandmother's from Kansas. What we measured in this study was it's called hyphal density. You can think of this as if you took a teaspoon of soil, how many threads of arbuscular mycorrhizal fungal hyphae, living hyphae, would there be in that teaspoon of soil? And so scientists have measured this all over the earth. And what they've done is they have collected soils in different environments, ecosystems under different host plants. We went through around 3,000 studies in 11 different languages, built this database. And from that, we used AI satellites to build a global map. And so what we get out of it is a spatial understanding of how arbuscular mycorrhizal hyphae are distributed across Earth ecosystems. So I zoomed into Kansas because you see there's kind of this like refuge, like it's like a refugia for this bright area where you have high density of arbuscular mycorrhizal fungi. So for example, this random pixel I click on here, we predict there's almost 8 meters of mycelium in a cubic centimeter of soil. I'm sorry for those in North America. I'm actually becoming Dutch at the end of the month, so I've forgotten everything in meters and in those units. But the point of it is now we have a spatial understanding of the infrastructure that is built to complete a resource transfer globally of plant carbon to fungi. These are the two major worlds that we've been merging, both this micron scale and the macro scale. So I found these helpful to start. I'm going to stop sharing my screen, but I think these are helpful, Jack, to frame the rest of our conversation.
Yeah, thank you for sharing that. And yeah, we'll link the link to both the paper as well as that visual tool in the description of this video. But yeah, thank you so much for sharing that. I think it's really great to have these visual tools that are accessible to the general public. So do you want to tell me a little bit about your scientific background and about how you got interested in this problem of the global distribution of AM fungi? Because that's a really big task to take on.
Definitely. So I guess my scientific background has been looking at microbes for a little over 10 years. I've mostly been interested in them because of, I hate the phrase ecosystem engineers, but they are ecosystem engineers. They're so vital to so many parts of planetary health and function, both in soils and in other environments. So I first started studying soil microbes in Austin, Texas, looking at soil carbon cycling, and then I moved to New York City subways and the atmospheric microbiome. And then I realized the soil is where we know the least. It is one of the least explored ecosystems on Earth, but at the same time, one of the most biodiverse and complex over both space and time. So then I look for what organisms are the least studied, and this was one of the most open questions about mycorrhizal fungi. So I was really motivated by the symbiosis in this. My PhD is on the evolution of cooperation and conflict in the plant microbial symbioses. And mycorrhizal fungi are just the least mapped, the least understood for a variety of reasons. People look at above ground life, or they use tools that are very focused on looking at soil bacteria, which are much more easy to sequence and culture, relatively easier,
Yeah.
still quite difficult. These were the motivating factors at looking at mycorrhizal fungi, and in particular, arbuscular mycorrhizal fungi. Love the ECM as well, but the focus on arbuscular was very practical in that they form symbioses with most plant species and are arguably the most common plant fungal symbiosis on Earth.
Yeah. So, when did you realize that this gap in understanding, where and how much AMF there is on Earth was something that needed to be addressed? And yeah, what brought you to taking on that challenge?
I can give you the fun answer as well as the real academic answer. So the
Do
fun
both.
answer, I'm going to do both. The fun answer is so at the VU at my university, I had an office opposite my advisor Toby Kears, and I never really went in for questions because I didn't really have many questions. But then one day I only went into her office one day for a question. And I went in and I asked her something about rhizobia. And then I was also asking about mycorrhizae, just how much are there on Earth? I just wanted to know out of pure curiosity, how much are there? So, for example, in 2010, a researcher had mapped how many trees are on Earth. I was curious about the same question. How many hyphal threads are there? And she said, well, yeah, we all want to know. That's the question. And so over time, working with my colleagues, I figured out people were building these databases of hyphal density. And once I learned they're spatial, my background in spatial mapping told me we need to make a map of these fungi. So that's the more fun side is I asked my advisor and they really said no idea. And then I find out my colleagues are already building parts of these databases, led by Corentin Bissot, who is one of the other co-first author. He's the other co-first author on this, and then the third author, third name, first non-co-first author, Rachel Cargill.
Yeah.
We're building these databases, and then I helped expand it. Um, the more practical answer is we are addressing a very large gap. So, for example, with climate change, there's growing interest in understanding the flow of carbon above and below ground. I say flow because I don't really like the idea of carbon stocks. It's managing flows above and below ground and the rates of these pools. And if you look at these things called ESMs, which are Earth system models, which try to understand the flow of this carbon, the difficulty is almost all of them could put microbes in one big black box, and it just says microbial carbon, and it's all controlled by carbon use efficiency. And of course, there are mycorrhizal scientists who said, well, this is absolutely not good enough. And they've built mycorrhizal models, and these have been foundational at us trying to identify what are their next steps. And we saw in these Earth system models that include fungi, mycorrhizal fungi, they rely entirely on plant distributions. So they say this is the location of a plant host that forms ectomycorrhizal associations. We expect this to be the role of carbon in it. But at the same time, we have emerging evidence that the distribution of above ground life may not be reliable for what you find in soils. So also last year, um, yeah, 2025 was last year. We published a paper in Nature about the biodiversity hotspots of AM and ECM fungi. And we find for AM fungi that largely the distribution of plant life above ground does not track what you see below ground. So this was one of the big motivating factors was we saw what you see above ground is not always a reliable indicator below ground
Yeah.
of biodiversity and species distributions. Because of that, we thought that we needed to develop a spatial model that is functional. So it's about the function of these organisms, the density of hyphae they grow every year, and it's important to note that our model is also limited to living hyphae. So the data that we had coming into it was all on living biomass estimates. So that's why we were very motivated both on the more fun side as well as practical. We need an advancement in the field. Those were our motivations.
Right. Yeah. And you mentioned how th this earlier work that someone did on mapping out, how many trees are there on Earth. So you had, it sounds like a similar problem to assess how much AMF there is, but these organisms are a little different in that it's really difficult to count individuals because they are these networks. Their bodies are a little hard for us to comprehend. So did that present any special issues in quantifying how much AMF there is because they exist as these networks as opposed to individuals? So do you want to talk about maybe any of the challenges that that presented or any other unexpected challenges in this work?
Definitely. So, I mean, there are many challenges in trying to do any type of spatial work. One of the most difficult, yeah, I guess I will split this into two types of challenges. One is spatial and one is non-spatial. The spatial challenge is that not that they're different networks, and that's important to mention here. So we estimate very long length. If you add up all of the hyphal threads end to end, it's around 110 quadrillion kilometers, about if you round 1 billion times the distance of the Earth to the sun. Our analysis is not saying this is one long continuous network. These are collections of individual networks. While you can have one plant that's connected to the same fungal species as its neighboring plant, we're not saying this is one massive network. One challenge has been in doing this analysis is that the data was collected very randomly across the Earth. It's unevenly distributed. So we had to correct for this. And that's something we were able to do in our model. And then the second part is it's really hard. So there were two parts of this analysis. Maybe I'll zoom out and then explain the second difficulty. We did not really use complex math in this. What we used was a very simple equation that Archimedes came up with. And so it's estimating the volume of a tube. So for this, we need to have two numbers. So its volume is equal to pi times radius squared times length. And this helps us get to biomass. We first need to know how long are these fungal networks on Earth. And then we also had to know how wide they are. So the first part about the spatial modeling and their density is about how long these fungal networks are. The second difficulty is just how wide are hyphae? And that sounds like it's a non-trivial problem. I always describe it as saying, imagine it's winter and you're lying underneath a tree and you're looking up at that tree crown. So there are no leaves on it, assuming it's deciduous. What is the average radius of of a branch on that tree? What's the average radius across that entire tree crown? It's quite difficult because you have some short, fat branches, but you also have some very long, skinny ones.
Yeah.
So this was really a difficult question to answer. So for that, we actually had to build a robot. Its name is Prince. Prince takes photos at, I believe, every 2 hours on petri dishes, where one half of it, it's cut in half. The top part of it has a mutated carrot root most often. And then the bottom part is growing fungal network. And so we're able to track the growth of these fungal networks over space and time. So we track about 500 million individual fungal nodes at any specific moment. And by tracking this, we were able to then estimate using a specific type of math called radius effective. We were able to figure out how wide are these networks. So we had to deal with two major difficulties to understand how heavy are the fungal networks on Earth. First, we don't have a complete understanding of their distribution and it's unevenly sampled across Earth. So we had to create predictive models of its length, as well as understanding the width of these hyphae. By combining these two and these two difficulties, we were then able to estimate how much these fungal networks weigh. Those were the two difficulties we had to deal with.
Yeah. So yeah, you mentioned using that robot to estimate hyphae radius, and that if people want to go and look through that visual story, or this is also
Yeah.
described in the paper in more detail, that whole process is kind of laid out there. And I was wondering, you mentioned, I think you guys did this estimation on four different AMF strains, correct?
So yeah, I'm actually pulling up the robot right now so we can
Yeah.
look at it while I explain this part. So but yeah, I encourage you to please look through this video and the different parts of the story we've built. We built this both for non-academics was the target audience, but I hope that people who are still academics and deep in the field are interested in this. So this is showing prints. And so these are examples of the fungal networks growing over space and time. And we're able to capture quite a lot of information out of these. So yeah, you can see there's like the plant on one side, and on the other side is a fungal network growing. I'm sorry, Jack, could you repeat your question? I was trying to get the video up.
Oh, well, what I was going to ask is you also mentioned some of Spun's work in mapping global biodiversity of AMF, and they've amassed these really large databases looking at, what AMF taxa are where, basically. So, do you think that integrating that data and maybe capturing more like species specific or strain specific information about hyphal morphology would help improve this model that you've put forward in this paper?
So I'm happy you asked this question. Our goal with this study was to estimate global biomass. And with that, there's the law of large numbers. You can have a bit of uncertainty and error with what specific species you're looking at. So it's important to say our biomass estimates, not our density, but our biomass estimates are best seen at aggregated scales, so at a region or global scale. If you want to turn something like a density measurement into a biomass estimate, for sure, local information on community composition and hyphal traits is vital to do that.
Yeah. And then one other thing you mentioned when talking about the challenges is this uncertainty piece. And this is part of the paper where you kind of describe areas where sampling effort is maybe different. And because of that, it seems like certain regions, it's more difficult to make these estimates than others. So now that you kind of have this resource and this map where you can see areas of uncertainty, what do you hope that the mycorrhizal research community does with that information, and how do we, broaden that sampling effort?
So I see uncertainty as a call to action. What a map of uncertainty, a spatial map of uncertainty is a treasure map, right? It tells you where to go next. And I find that extremely charismatic in that you can say, well, we feel confident with a prediction here, but maybe 10 kilometers away, we're less confident. And this is somewhere we need to ground truth it. We need to produce data to validate our predictions. And this is something that I find really, really difficult to communicate, but it's important, the importance of scale. So again, as I mentioned, aggregated scales are what we're testing in this data set. But as you move into smaller scales, this is where action needs to happen. So I hope that this type of research stimulates mycorrhizal researchers to go in their local ecosystems and look at things like hyphal density. When you when you look at areas on Earth that are uncertain, the models predict they're largely in places far away from where samples were collected. Very different environments, very different ecosystems, which makes sense because they're not in the model's training data. So these are the areas that need to be most urgently sampled. They do largely overlap with areas where we also were lacking data on fungal biodiversity. I published a paper last year showing that 70% of Earth's ecoregions have been unsampled for AM fungi. It's quite similar for AM fungal biodiversity. It's quite similar for hyphal density. So I really do encourage researchers to go out and collect more data on this. That's how we got the data to build this first map. So I'm
Yeah.
very excited about validating it. The last part I'll say about this is don't throw away the good old tools. Everyone is so focused on eDNA and using new tech to measure these fungal networks in hyphae in soils and biodiversity. This relied on the easiest, depends on how much time you have,
Yeah.
cheapest, one of the cheapest ways you can measure mycorrhizae in Earth's ecosystems. You essentially need a soil core and a microscope and time. So if you look at the global distribution of where these samples have been collected, they're actually in countries that may not have as easy or accessible access to things like DNA sequencing. So I really encourage mycorrhizal researchers to try to pair together information on hyphal density with eDNA, for example, and we can start to link together functional information with community composition. So yes, many uncertainties remain spatially, but to fill in these gaps, we need to start linking together classical ecological sampling methodologies also with new technologies.
Yeah. And then in the paper, at one point you mentioned maybe the need for increased standardization of the way these results are reported or the way the data is collected. Do you want to speak about that a bit as you kind of put out this call to researchers to broaden this database?
One of the most important parts of this call to action is please put the GPS coordinates of your samples in your studies. We
Okay.
had to zoom into so many locations on Google Maps and try to figure out where sample sites were located and georeference maps onto Google Earth.
Yeah.
Just open data is so important. You publish your data sets with coordinates and as much metadata completely done as possible. This means that your data is also being able to be used for others as well. I mean, it's a lot. Like, think about how much work you do to collect a soil sample, making sure that that data is out there and public, and often it's funded by public money, taxpayer dollars. I think this is a very important part of the research. So, one of my calls of action is to standardize the way people are reporting their data. And ideally, in the future, we would standardize the way we start collecting soils. But I understand that this varies based off of study design and research question. But we saw so much variation. Sometimes people collect 3 replicates per site. Sometimes they collect 50. Sometimes they collect only 2 grams of soil. Sometimes they collect the whole soil core. That part I think is less standardizable. But what is standardizable is how you report the data with its metadata.
Gotcha. Okay, so just to pivot a little bit, um, you obviously this article is published in Science, which has a very wide readership, wide academic readership, and then you've also put together these really great visual tools, and it seems like that's a common theme with Spun is public outreach. So through all of those ways to communicate this work, who do you hope that this reaches that might not have been thinking about mycorrhizal fungi before this work?
I'm going to answer this in 2 ways. I have 3 audiences. One is the intended academic audience, and that's why we're doing this call, right? This
Yes.
is largely going to be listened to by academics. I want this research to um, in the more fun way. I wanted to stimulate a sense of wonderment with people. I feel like in the world of the current world we're living in, it's really difficult to feel joy or wonderment or excitement, but we've now just started mapping a vast part of Earth's circulatory system that we had no idea existed. And it kind of puts a middle finger up to everything humans think they know about life. They don't follow above ground life very well. Sometimes they can have meter long cells that have 2 types of genomes in them. There's no part for central information processing, but they enact very complex trade decisions.
Yeah.
So the first is a sense of wonderment about how much there is left to explore, largely also in Earth soils. And then the very practical one is I want this data to be communicated and handed off to decision makers. So that's one reason we developed this interactive online tool. And we've been putting so much information with work in with press. I have to shout out Magda at Spun, who's just done an incredible job. Is we want this to be a conversation starter about, hey, what is the health of a mycorrhizal ecosystem in our region or our soil? This is, I think, one of the most important parts of this is figuring out how can we learn how to map and protect these fungal networks. to that effort. Spun is interested about we're interested in getting these data into the hands of different people. So it's going to be presented at COP in Mongolia. That's going to be coming up, as well as um, coming up soon in Quito. I will be presenting this work, but we're also having a workshop there called with underground advocates, is what it's called. So these are going to be a team from NYU called Moth, who is going to be More Than Human Life, Moth, who's going to be coming in, and we're going to try to equip mycorrhizal scientists with the information to talk to decision makers with a bit of a legal background about the health of their mycorrhizal systems. So there's both the sense of like, I'm still a kid and I want to know fun science facts, but also the urgency there is around protecting mycorrhizal systems and enabling researchers to act on that locally. So it's both.
Yeah, that's awesome to see mycorrhizal research kind of reaching across disciplines and reaching people even outside the scientific field. Because I feel like having some of these statistics that you estimate in this paper, like how long is all the hyphae on Earth, or how much does it weigh? Those are very useful for us as researchers to be able to convey kind of the magnitude and the importance of these organisms that we spend so much time focusing on. One thing I was wondering is, before starting this project, would you have expected those numbers to be as large as they are, or what was your best guess before starting to peel into this data?
My best guess. So I expected big numbers. I
Uh,
mean, my background in addition to evolution is really statistics. And like I became known in my group as the guy who, if you just want to estimate a number, so I think a lot about just what are our scales of magnitude. I don't care if the number is 6 or 8 or 10. I want to know, is it 10 or 100 or 1,000? And so doing some back of envelope calculations, we expected around this order of magnitude for length.
okay.
But then when we finally started estimating how much these fungal networks weigh, like we reran the numbers for like 2 or 3 weeks, like it felt wrong. So 110 quadrillion kilometers is our global estimate, plus or minus the error term, which is around one order of magnitude smaller. But they weigh around 5 times the biomass of all humans on Earth. That's really lightweight. And that was something at first when we were analyzing this and talking with the co-authors, we were concerned about putting that out because a lot of the conversation about Earth's ecosystems is focused on how much biomass is there, how much do they weigh, and often this
Yeah.
comes from our human above ground bias of looking at plants and saying that tree is carbon, we need to think about how much it weighs. But the interesting part of our story is not the weight, it's the weight given the length. These networks are, we estimate, around 50 times longer than plants' fine roots on Earth. So they're so long and so thin, they can get deep into Earth's soils where they're able to deposit carbon, for example, through exudates, or their biomass can turn over. Biomass can also be released back to the atmosphere through things like priming and respiration. But it's the fact that they're a long, thin infrastructure that can increase, for example, a root's foraging area. This is their contribution to Earth's health. It's not how much they weigh, it's the fact that they're this underground highway system. So this was really difficult in the beginning. We didn't know how people would receive this finding or how to communicate this well, just because of like the zeitgeist around carbon.
Right. Yeah, so you actually expected the biomass to be a little higher. Okay.
Yeah, I I mean the first thing we built the numbers, length and biomass, and there was a few weeks where I was just like, how do I convert this to something a human can understand? Because I don't know what 1.0 times 10 to the 17 is. I have no concept of what that
Yeah.
is. And so we first started comparing, for example, length to all the roads in Germany, the length of all the roads in Germany, and then it's like 38 billion times that. I'm like, well, I have no idea what that means. So the numbers, length, we had to go astronomical. And then so I immediately thought, all right, let's compare the weight to the weight of the Amazon rainforest to all the carbon in the tundra. And it kept coming out to such small numbers.
Yeah.
And so it was just very difficult to grapple with the fact that we don't think they weigh a lot. But I mean, if you've ever looked at mycorrhizal fungi under a microscope, or AMF in particular, they're just so lightweight and so thin
Yeah.
that, I mean, of course it makes sense. But again, I was just being very influenced by the bias that I think they should weigh more.
Yeah. But yeah, I think it's important to remember, like you showed at the beginning there, that video of the flow throughout fungal hyphae. These AM hyphae are super dynamic, so they yeah kind of exist in this state of change and are yeah moving carbon around in maybe a way that could be a little underappreciated. Okay, so to kind of wrap up our interview here, we always like to ask our participants, what are you focusing on next, and where do you hope that your research can take you?
Well, that's a secret. I can't tell you. I'm kidding. I'm kidding. I'm still very interested in understanding the distribution of mycorrhizal fungi. So both biodiversity and their biomass and their structures. So this I think will be coming out before ICOM or at least for this keto presentation, but I've been working on also mapping AM fungal spores and comparing this to the distribution of hyphae. So this is what I'm most excited about right now.
Awesome. And are there any other parting words that you want to have for our audience?
Protect the fungi. Please explore our mycorrhizal infrastructure map and get in touch if you're interested in learning how to use these types of data and hand them off to decision makers.
Great. Yeah, and we'll put links to all the resources that we talked about in this video in the video description, as well as maybe some contact information for you, Justin.
Definitely.
All right,
Thank
so thank
you.
you for joining us for Mycorrhizas, Justin, and thank you to the audience for listening.
Thank you all.